black box inside the cell. Matsui and his colleagues loved to watch frog eggs. They enjoyed watching the fertilized eggs divide over and over again. The new cells divided so quickly they didn't even have time to grow up before they divided again. In fact, they did not need to spend time in G1 or G2 phase at all. Everything these cells needed came from the egg they first divided from. Those are cleavage divisions, noted Matsui. He thought about how many divisions had happened. I wonder what makes them divide, he thought. He remembered that before the frog egg matured and was fertilized in the first place, it was just an oocyte, and it was not dividing. Why is it dividing now? Somebody was stopping it before. Matsui and his colleagues set up an experiment. They took an extract of cytoplasm from a frog egg in the M phase and put it into an oocyte. Dear me, exclaimed Matsui, I can see a spindle forming. The oocyte is maturing. Now the oocyte was going into M phase, just like the phase the egg was in. What happens if I use cytoplasm from one of the cleaving eggs in interphase? That one has already gone through M phase and may do so again. Will something different happen? Something different did happen. That is to say, nothing happened at all. How very curious, thought Matsui and his team. There must be a factor in the cytoplasm of a maturing egg. There must be a maturing factor in the cytoplasm that makes cells mature. And that was a very clever name for this curious thing, or so we thought. Perhaps too clever, so we shall just call it MPF for short. Chapter 2. Tim Hunt liked to look at clam eggs instead of frog eggs. He was particularly interested in the amounts of M. cyclin because sometimes there are more and sometimes there are less. Sometimes Hunt would see on his cell cycle chart that these proteins would rise, and then fall. This puzzled Mr. Hunt, and he was determined to figure out just what was happening in those little clam cells. Why are there so many of you, he asked the M. cyclins, but they did not answer, as M. cyclins aren't the chatty sort. He returned later to find that almost all his friends, the M. cyclins, had gone away. Where did they go? He pondered. Later, he was relieved to see his friends were slowly coming back around. Hey, said Hunt indignantly, what do you think you're doing stuck to that MPF? Those silly cyclins were partnered up with MCDKs. They like to stick together, it seemed, and get work done. Like cooperation and teamwork, you may ask? Yes, perhaps something a little bit like that. After those partners were found, it was easier to find other kinds of cyclin and CDK partners. They rise and fall on the chart just like M cyclins, but at different stages in the cell cycle. Chapter 3. Eggs and clam eggs. Lee Hartwell and Paul Nurse liked to look at yeast. Lee Hartwell looked at baker's yeast and Paul Nurse looked at fission yeast. Instead of looking at protein levels, they looked at their genetics. I can see a lot of genes that control the cell cycle, boasted Hartwell. I can see them too, and better ones probably, Nurse might have said. He might not have too. No, I don't think so, Hartwell might not have said in return. I doubt either one of these men were belligerent. However, they both found a lot of these genes by looking at belligerent cells. Mutants stuck at stages in their cell cycles refusing to do as they should. Nurse and his colleagues said to each other, look at all these different kinds of things that have the same kind of cell cycle control. Frogs, clams, yeast, maybe even people like you and me. They thought they should test the idea, and so they did. They took a yeast with a bad CDK that wouldn't divide, and they gave it a good CDK from a human cell. Amazing. It worked. Chapter 4. Matsui, Hunt, Hartwell, Nurse, and all their helpers all taught us a very important thing about the cell cycle. All eukaryotes, big and small, use the same molecules to control their cell cycle.